Hello, welcome to the Clinical Care Options Neuroscience podcast series called The Future of Brain Cholesterol Turnover for Neurologic Diseases. This podcast series is supported by an independent educational grant from Takeda Pharmaceuticals USA Incorporated. The focus of this podcast is entitled Identifying the Emerging Role of Brain Cholesterol Turnover in Neurological Diseases like Epilepsy. I will be your host. I'm Dr. Jonathan Halford, and I'm an adult neurologist and epilepsy specialist at the Medical University of South Carolina in Charleston, South Carolina. Today, I have the pleasure of speaking with Steve White, who is a pharmacologist and a professor in the School of Pharmacy and co-director of the Center for Epilepsy Drug Discovery at the University of Washington in Seattle. Before we begin, I have a quick housekeeping note. This episode is accredited for 1/2 of an AMA PRA Category 1 credit, and also for ANCC contact hours. After listening to this podcast, please click on the link in the show notes to take the post-test and earn your CE or CME credits. All right, let's get started. Dr. White, it's a pleasure speaking with you today. Can you tell me a little bit about your background and current research?
Certainly. First, Dr. Halford, thank you very much for having me with you today. And, by way of full disclosure, as you mentioned, I'm a pharmacologist. My patients, walk on all fours and have red beady eyes. I have been, over the last 40 years, conducting preclinical research, largely focused on the early identification and characterization of investigational anti-seizure medicines. First at the University of Utah, where I spent 35 years. In the last 15 of those years, I served as the director of the NINDS-sponsored contract there by the Epilepsy Therapy Screening Program, which really has been the stalwart, if you will, of epilepsy drug discovery over the last 5 decades almost. In addition to using various animal seizure and epilepsy models for characterizing the potential anti-seizure activity of these investigational drugs, our work has focused also on the development of new models that may be used and more clinically relevant to some of the different seizure types that patients deal with. In 2016, moved to the University of Washington, and a lot of the same work that I did at Utah is ongoing in our laboratory in the newly formed Center for Epilepsy Drug Discovery here at the University of Washington.
All right, great. Well, it sounds like you have a very strong background in epilepsy, animal models, and basic science research. We'll be talking today about cholesterol and its role in the brain. When I think of cholesterol, I think of atherosclerosis, which causes heart attacks and strokes. How is cholesterol involved with brain function?
You're absolutely right. I mean, we've all been taught that, cholesterol is sort of a precursor to arterial disease and potentially stroke, and that we should be sort of maintaining a proper diet to keep cholesterol levels. But I think it's also important to remember that cholesterol is a crucial component of every cell membrane throughout our body. And it's also important to note early on that cholesterol is really poorly absorbed when eaten. We don't really get it from our diet. And our body really makes the majority of our cholesterol from other fats, which are consumed in our daily diet. Also, it's interesting for the purpose of this conversation that cholesterol is found in a much higher concentration in the brain and the spinal cord relative to the rest of the body, and it plays a critical role in maintaining proper brain function.
All right. Well, you mentioned that cholesterol is found throughout the body. What's its function in cells?
Well, as mentioned, it's abundant in plasma membranes in all cells, and it really functions to regulate the property of cell membranes, such as membrane fluidity, thickness, and permeability. And it serves really as a glue for the organization of the membrane microdomains called lipid rafts. And lipid rafts are dynamic sort of assemblies of various proteins and lipids that float freely within the bilayer of cellular membranes, but they can also cluster to form larger, more ordered platforms. Cholesterol concentration at the lipid rafts critically regulates ion channel permeability, signal transduction, synaptogenesis, and synaptic vesicle dynamics. And it's in this role that it really can modulate neuronal excitability and inhibition through its effects on ion channel permeability and signal transduction.
So it sounds like in the nervous system that cholesterol is important for cell signaling then, right?
That's right. In the nervous system, it's found in high concentrations, higher than the rest of the body. And to that point, the brain contains almost 25% of the body's total cholesterol. And you might ask why, and largely because it plays an important role in brain and nerve cell membranes, particularly the membranes of glial cells and neurons of gray matter. In this respect, it makes up a large part of the myelin sheaths, which, as we know, are a highly specialized plasma membrane that wraps around axons. Myelin acts as an insulator, and through the saltatory conduction that, we might remember from our basic physiology, these insulators and the nodes of Rambier play an important role for speeding up the conduction of action potential. Additionally, the brain uses cholesterol to synthesize neurosteroids. And neurosteroids are a neuroactive neuromodulator, if you will, which rapidly can alter neuronal excitability through their interaction with ligand-gated ion channels, and particularly the chloride ion channels that are gated by the inhibitory neurotransmitter GABA. And to this point, there are actually two new therapies in development for the treatment of epilepsy that target these neurosteroid receptors. So I think, we're going to see a lot more development of drugs like the neurosteroids that really we learned a large part about their function by looking at cholesterol metabolism and the products of cholesterol metabolism.
So it sounds like cholesterol is very important for the brain. And if we don't get it from our diet, does the brain make its own cholesterol?
Yes. The majority of the cholesterol in our body and brain is locally synthesized. And it can be synthesized, obviously, in the periphery, but cholesterol circulating in our blood cannot pass the blood-brain barrier. But there are varying cholesterol synthesis capabilities in the different brain cell types. And importantly, I think it's worth mentioning that the brain cholesterol has a long half-life, and the estimated half-life of cholesterol in the brain is really years, whereas in the systemic circulation, it's on the order of days. So most of the cholesterol that's in the brain has a very low turnover rate, and the cholesterol that is synthesized in the brain, largely in astrocytes, can transfer to neurons and oligodendrocytes. And in the oligodendrocytes is used to synthesize the myelin that we talked about to make the myelin sheaths. And in this regard, because we don't get it from our food, it's not considered an essential nutrient.
All right, thanks for that information. So what is the metabolic pathway for the synthesis of cholesterol?
Well, it's a complex multi-step process, and there are multiple pathways for synthesizing cholesterol. And it starts really with the conversion of acetyl coenzyme A to acetyl coenzyme A, 3-hydroxy, 3-methylbutyryl coenzyme A, or HMG coenzyme A. Subsequently, mevalonate, and these are catalyzed respectively by a couple of important enzymes. One of the enzymes here is something called HMG coenzyme A synthase 1 and HMG coenzyme A reductase. And the reason this is worth mentioning is that the rate-limiting step in the biosynthesis involves this HMG coenzyme A reductase. And this is the target of the widely acceptable cholesterol-lowering drug, collectively known as statins, which are used to treat dyslipidemia or hypercholesteremia. And to this point, interestingly, we've in recent years have actually found that the statins can be neuroprotective. And whether it's through their effect to decrease cholesterol metabolism or through their anti-inflammatory action that can result from both direct actions, but also a decrease in cholesterol can result in a decrease in inflammation.
So if the glia make cholesterol and it's for other brain cells, then how is cholesterol transported in the brain?
Well, as you pointed out, cholesterol is primarily synthesized within astrocytes or one of the glial cells, but used largely by neurons and oligodendrocytes. And so, as you alluded to, it needs to be transported to the appropriate cell type. And this is done by a unique lipoprotein called HDL-like particle, which is similar in size to HDL, which, we all hear about every time we get a lipid profile done in the doctor's office. So it's similar in size to HDL in the plasma and made by the astrocytes. And these HDL-like particles contain a component called ApoE. And the main function of ApoE is to regulate lipid transport between neurons and glia, as well as the metabolism of lipoprotein-bound cholesterol. It's a sort of tight, well-regulated process of synthesizing and transporting cholesterol to get it to the right place.
So what are the factors that regulate how much cholesterol there is in the brain? And how does the brain get rid of cholesterol that it doesn't need?
In the blood, the half-life of cholesterol is only a few days, but cholesterol turnover in the brain is extremely slow, and the half-life is in years. The amount of cholesterol, therefore, in the brain is mainly determined by how much is synthesized, because the breakdown of cholesterol in the brain is so slow, not much new cholesterol is actually needed. However, if it happens that too much cholesterol accumulates, the brain does have a mechanism to break it down or to metabolize it. And conversion of cholesterol to oxysterol is a major excretion pathway in the brain. And this involves hydroxylation of cholesterol to 24-hydroxycholesterol, which is catalyzed by the enzyme cholesterol 24-hydroxylase. Most of this enzyme is present in neurons, not astrocytes, and it is particularly found in certain neurons such as pyramidal cells, the cortex, Purkinje cells of the cerebellum. And its presence there suggests that these types of neurons may be particularly sensitive to cholesterol levels.
So is cholesterol metabolism involved in the pathogenesis of any diseases?
As we've learned over decades now, cholesterol metabolism is a delicate and it's a complex process. Cholesterol is required for several physiological functions of the brain, including synaptic development and synaptic transmission. As such, it sort of reasons that alterations in cholesterol metabolism could have profound effects on brain dysfunction. And one disease which clearly involves cholesterol is Niemann-Pick type C. And in this disease, there is a mutation in the Niemann-Pick C1 gene, which normally produces a protein that transports large water-insoluble molecules like cholesterol through the cell wall. The disruption of the transport system by this mutation results in the accumulation of cholesterol and glycolipids in lysosomes, or those organelles that break down excess or damaged cell processes. Niemann-Pick type C protein deficits result in impairment in intracellular cholesterol trafficking and the dysregulation of cholesterol biosynthesis. In addition, disruption of cholesterol homeostasis also associated with a deregulation of autophagic activity and early onset neuroinflammation, which, as we know, may contribute to the pathogenesis of Niemann-Pick type C disease, including the severe progressive neurological dysfunction associated with this disease. In addition to Niemann-Pick type C, abnormal cholesterol metabolism has been associated with the pathogenesis of other neuronal diseases, including Alzheimer's disease, Parkinson's disease, Huntington's disease, and epilepsy.
How is cholesterol metabolism involved in Alzheimer's disease?
Well, the association between cholesterol metabolism and Alzheimer's disease is a bit confusing and can be conflicting. As we know, Alzheimer's disease is characterized by beta amyloid or A-beta peptide aggregation and senile plaques. And some studies have suggested that high cholesterol in middle age could be a risk factor for Alzheimer's disease development and dementia. Preclinical studies in rabbits have shown that a cholesterol-enriched diet can induce Alzheimer's disease-like pathology, including increase in A-beta, tau phosphorylation, and oxidative stress. The plaque formation in Alzheimer's disease, as we know, results in damaged neurites and synapses, and as a result, disrupts neurotransmission, which can certainly contribute to cognitive decline and dementia. Importantly, A-beta is formed by the successive cleavage of amyloid precursor proteins, or APP, and high levels of membrane cholesterol leads to the incorporation of A-beta into the membranes and also increases the amount of calcium inside a neuron, which can be toxic and contribute to neuronal cell death. As we mentioned earlier, ApoE is an important component in the lipoproteins which transfer cholesterol. And ApoE has three isoforms known as epsilon 2, epsilon 3, and epsilon 4. A person who carries the epsilon 4 allele in his or her genome is known to be at risk of developing Alzheimer's disease. And ApoE can bind to the numerous receptors on cell surface for the delivery of cholesterol, and also to the A-beta peptide, which initiates the cascade that leads to neurotoxicity and to the neurodegeneration seen in Alzheimer's disease. Interestingly, studies have also shown that a diet containing high cholesterol enhances the level of A-beta peptides, and concentration of serum cholesterol rises approximately, or has been reported to be approximately 10% higher in Alzheimer's disease patients than that of healthy individuals. Now, we haven't talked about the metabolites of cholesterol metabolism, but high cholesterol levels indicate the possibility of high levels of oxysterol, 27-hydroxy cholesterol, and 24-hydroxy cholesterol, as well as 25-hydroxy cholesterol. The reason this is important is that these oxidized sterols have toxic actions that can include an increase in oxidative stress, a reduced glutamate reuptake by decreasing excitatory amino acid transport on astrocytic membranes, thereby reducing the reuptake of glutamate from the synaptic space, and also 24-hydroxy cholesterol has been shown to be a direct allosteric modulator of the NMDA glutamate receptors. Now, what's interesting about cholesterol levels in Alzheimer's disease is that, as I just discussed, high levels in midlife have been associated for an increase in Alzheimer's disease. But in contrast to midlife, some studies have actually suggested that low cholesterol or hypocholesterolemia can contribute to dementia later in life and perhaps Alzheimer's disease. And although not definitive, it's thought that low cholesterol is the result of a reduced ability to synthesize enough cholesterol to maintain good membrane stability, that thereby impacting synaptic function. So I think there's a lot more to come on the role of cholesterol in Alzheimer's and the interplay between, membrane synaptic function and the various transport processes.
That's very interesting. Sounds like there's a lot of connections there, and it sounds like it's quite a complex and evolving field. So how's cholesterol metabolism involved in Parkinson's disease?
Parkinson's disease, like Alzheimer's disease, is a neurodegenerative disease, and its hallmark is the accumulation of alpha-synuclein and the formation of Lewy bodies. The clinical symptoms of Parkinson's disease result from a loss of dopaminergic neurons in the substantia nigra and the accumulation of alpha-synuclein and the formation of these Lewy bodies. And it's been reported that high lipids can increase the accumulation of alpha-synuclein, which is one of the main constituents of Lewy bodies, by stimulation the formation of alpha-synuclein aggregates. Thus, in this way, you can perhaps see how a high level of cholesterol and its oxidized product, or oxysterol, are thought to play a crucial role in the development and severity of Parkinson's disease by facilitating this alpha-synuclein aggregation. With this in mind, some have even considered that high cholesterol levels and oxysterol levels could serve as potential biomarkers. With that said, I think it's important to note that Parkinson's disease, like Alzheimer's disease, there's still a lot to learn, and there's really not full agreement on what role cholesterol may play in Parkinson's disease pathogenesis.
All right, well, thanks for explaining that. So how is cholesterol involved in Huntington's disease?
Well, Huntington's disease is caused by an abnormally high number of CAG repeats in the Huntington gene, and it's characterized by the degeneration of neurons in the cortex and striatal regions, which can lead to cognitive issues and a characteristic movement disorder, including dyskinetic movements. Cholesterol metabolism is impaired in Huntington's disease in proportion to the length of these CAG repeats, and it involves attenuated or reduced plasma cholesterol levels, reduced brain cholesterol synthesis, and cholesterol accumulation in neuronal plasma membranes. So much like Alzheimer's disease and Parkinson's disease, exactly how this abnormal cholesterol metabolism contributes to the pathogenesis of Huntington's disease is a bit complicated. It's a bit unclear at the present moment. What we have is an association, and clearly more research is needed to fully understand the relationship.
All right, got it. So how is cholesterol metabolism involved with epilepsy?
The link between cholesterol metabolism, the pathogenesis of epilepsy, has been recently described to involve the enzyme cholesterol 24-hydroxylase. This enzyme, as I mentioned a little earlier, catalyzes an important step in the breakdown or the metabolism of cholesterol through the hydroxylation of cholesterol to oxysterol. This is the major excretion pathway for cholesterol in the brain. And the possible link between cholesterol 24-hydroxylase enzyme and epilepsy was discovered when it was observed that in a transgenic mouse model of Alzheimer's disease, the sudden death caused by convulsive seizures was prevented by a drug called soticlestat. And soticlestat is a first-in-class inhibitor of the cholesterol 24-hydroxylase enzyme. Subsequent studies demonstrated that cholesterol 24-hydroxylase promoted the generation of oxidation products, which leads to oxidative stress and glutamate-mediated excitotoxicity, which, as we've come to learn, both of these are an important process underlying epileptogenesis, or the trigger or triggers that lead to the development of epilepsy.
So if soticlestat is a possible therapy for epilepsy, we don't have any treatments or any drugs currently involving cholesterol for Alzheimer's disease, Parkinson's disease, or other neurological disorders, though, right?
That's correct. Not at the present time. I think, it's important to know that through the research that's been conducted and understanding the metabolic pathway of cholesterol synthesis and metabolism, we've learned a lot about various targets that may be amenable to therapy development. And soticlestat is an excellent example of how that research has led to a potential therapeutics for the treatment of epilepsy. And I think it's reasonable to expect that with greater research and a greater understanding, other targets may become available for the treatment of Parkinson's disease, Alzheimer's disease, and perhaps Huntington disease.
So I understand that soticlestat is being studied in clinical trials for epilepsy. What types of epilepsy are being studied?
So at the present time, we don't know exactly what types of seizures and epilepsy soticlestat may be useful in, but it is being currently investigated for the treatment of developmental and epileptic encephalopathies, which include Lennox-Gastaut syndrome and Dravet syndrome, as well as CDKL5 deficiency disorder or cyclin-dependent kinase-like 5. Patients with these types of epilepsy usually have childhood onset seizures, developmental delay, cognitive disability, as well as a multitude of different seizure types that unfortunately are often non-responsive to the treatment that we currently have available. The hope is that soticlestat's unique mechanism of action will bring a new therapy that may help reduce the frequency and severity of seizures in these disorders. Whether or not it will be useful and effective in the treatment of drug-resistant focal epilepsy is yet to be known. The truth really lies in the results of ongoing clinical trials and these childhood developmental encephalopathies and future clinical trials that may involve other seizure types.
So are there any other compounds besides soticlestat that you're aware of that also affect brain cholesterol metabolism that might be tested at some point for neurological diseases?
It's a great question, and to be honest with you, I'm not aware of any other therapies currently in development that target cholesterol metabolism in a way like soticlestat and has the data to suggest that they may be effective in brain neurodegenerative diseases.
All right, great. Well, thank you very much, Dr. White, for discussing this with me today. That was very informative. And thank you to our listeners for joining us. As a reminder, to view the full program, call it The Future of Brain Cholesterol Turnover for Neurological Disease, a podcast program. Please click on the link to the show notes. And thanks for listening.
Thank you very much, John. Appreciate that.